The Lac Operon. The Lac Operon is a classic in genetics. In fact, around this time, all across the country, students are probably learning about the Lac Operon. And the Lac Operon never goes away. Just anecdotally, I teach a graduate class in applied DNA biology here, and we were just talking about Lac Operon Regulation a couple weeks ago, because some of the most tried and true and traditional recombinant DNA technologies and protein expression technologies that we use in biotechnology and in other laboratory applications rely on principles of the Lac Operon. So this isn't only important as an example of how a real gene is regulated in a bacterial cell. The Lac Operon really has its fingers deep in the life sciences in many, many different ways. So let's give a little bit of background on the Lac Operon first so that we have some context here. The Lac Operon is actually a series of 3 different genes that are all under the control of a single, what we refer to as a promoter. You can think of the promoter as an on-off switch for the gene. So when the promoter is active, these 3 genes are going to be expressed. And when the promoter is inactive or somehow blocked, we will not transcribe these 3 genes. Now these 3 genes encode 3 separate enzymes or 3 separate proteins, all of which together are needed in order to metabolize lactose as a sugar, as an energy source for bacterial cells. Lac Z encodes an enzyme that is called beta-galactosidase. Beta-galactosidase is a cleavage enzyme, and by that we just mean that it cuts the lactose sugar down into its 2 simpler sugar parts. That's the first step, really, of the metabolism of lactose, to cut it into smaller, more easily digested sugars. Lac Y encodes a protein called a permease. And if the term permease makes you think of permeable, you're right, because Lac Y encodes a protein that allows the cell to import lactose into the cell. So it's great that lactose is out there in the environment, but it's no good to the cell unless the cell can transport that lactose into its own interior. And permease is what allows the cell to do that. The last protein is an acetylase. And for those of you who are in organic chemistry, hopefully you're somewhat familiar with an acetyl group, a simple organic group. But acetylating lactose is important for some aspects of its metabolism. So what these 3 genes have in common is that all 3 of them are required for the cell to metabolize lactose as an energy source. And it makes sense, I think, that the cell will only need these genes to be expressed. It will only need these proteins if lactose is around. If lactose is not around, it makes no sense to express these proteins, because why would you make the tools that you need to metabolize lactose when lactose isn't present in the environment at all? But something very interesting and very fascinating happens as well. You see, bacteria doesn't really like lactose. It's not a favorite food of bacteria. What bacteria really like to eat is glucose. In fact, what our own cells really like to eat is glucose. Glucose is the most efficiently metabolized sugar for all living cells. Lactose is a more complicated sugar. It takes work to get lactose to be a metabolizable sugar. It takes effort on the cell's behalf. So the cell can actually make a choice between these 2 sugars. In other words, if both of these sugars are present in the environment, the cell will not eat the lactose. And by eat, I mean metabolize. The cell will make a cellular choice to metabolize glucose, even when lactose is present. That choice is not conscious, of course, because we're talking about bacteria. That metabolic choice is hardwired into the regulation of the Lac Operon. If neither sugar is present, yes, it's true that the cell is at some risk of dying, of starvation, but certainly expressing the genes needed to metabolize lactose will not help the cell survive when there's no lactose around to eat. Likewise, when glucose is present and lactose is not, there it makes no sense at all for the cell to express these genes, because 1, the favorite food is present, 2, the lesser food is not. So why express the Lac Operon, which you need for metabolizing sugars, lactose sugars, when there's no lactose sugar there to consume and your favorite carbon source is present? But when glucose is absent and lactose is present, There, the cell has another choice, an interesting choice. It can eat or metabolize its least favorite food, lactose, or die of sugar starvation. And E. coli is no dummy. E. coli will metabolize lactose when it is the only sugar available in the environment. So how the cell makes this metabolic choice of consuming and metabolizing lactose only when lactose is present and glucose is not is really the story of the Lac Operon regulation. So as much as the individual genes of the Lac Operon are important, I'll likely never mention them again, because everything that we focus on is going to be happening here at the promoter of the gene, where it is controlled. The last player in this system that belongs to the operon that we haven't talked about yet is Lac I. And the Lac I gene encodes a protein that we refer to as the Lac Repressor. And from that name alone, you can probably figure out that this protein is responsible for turning off the transcription of the lactose gene, of the Lac Operon. So normally, this repressor binds to the promoter and blocks transcription from happening. It represses the gene. But when lactose is present in the cell, some of that lactose is converted into a derivative sugar called allolactose. And I'm going to represent allolactose as a purple dot. Allolactose can interact with and bind directly to the Lac I repressor. And when it does so, the Lac repressor can no longer bind to the Lac Operon. In the presence of allolactose, the Lac I repressor is off of the DNA. When lactose is not around, allolactose cannot be made, and when allolactose cannot be made, the Lac repressor will be bound. So we might think that in the presence of lactose, when allolactose is being produced and the Lac repressor is no longer on the DNA, that the Lac Operon is on. But that's not the whole story. Because remember, we only want to express the Lac Operon when lactose is present and what? And when glucose is absent. You see, this promoter is a very poor promoter, and the enzymatic machinery responsible for transcription cannot bind to this promoter easily. We need an activator in order to turn this gene on. And that activator responds to glucose levels. When glucose levels are low, and this is true of most cells on this planet, that's a very dire cellular circumstance. Glucose is the preferred sugar by most cells. In fact, our brains can only metabolize glucose for energy, otherwise neuronal cells start to die. So glucose levels falling is a signal of a cellular emergency. And the cell makes a very important molecule in response to low glucose levels. It's called cyclic AMP, or CAMP for short. Cyclic AMP is an SOS molecule. It's an oh my lord, what's going to happen molecule. It's a molecule that sends a strong cellular signal that glucose levels are falling. So when glucose levels are low, cyclic AMP is made. And cyclic AMP is capable of binding to a transcriptional activator protein called CAP. But when cyclic AMP binds to CAP, this CAP protein binds to the promoter of the Lac Operon and recruits the cellular machinery for transcription. Now, CAP can only bind to the promoter when the Lac I repressor is absent. So let's think about this in terms of all of the cellular signals that we've discussed so far. We have cases where lactose is absent and glucose is absent. We have cases where lactose is absent and glucose is present. We have cases where lactose is present and glucose is present. And finally, we have where lactose is present and glucose is absent. If we think of these one at a time, we realize that when lactose is absent, allolactose cannot be made. And if allolactose cannot be made, the Lac repressor will be bound to the Lac Operon. When the Lac repressor is bound to the Lac Operon, the operon is off and lactose cannot be metabolized. Likewise, when lactose is absent and glucose is present, once again, allolactose cannot be made. And if allolactose cannot be made, once again the Lac repressor will be bound to the Lac Operon, and the Lac Operon will be kept off. That makes sense. Lactose is not present, and glucose is present. There's no need for the Lac Operon to be expressed. When lactose is present and glucose is present, now allolactose can be made. And that allolactose will bind to the Lac repressor and pull it off of the DNA. Now the promoter is available for activation. But glucose levels are high. If glucose levels are high, cyclic AMP levels are low. Without cyclic AMP, the cap protein cannot bind, and there is no activation of the Lac Operon. So even though the Lac repressor is absent, the absence of activation as well keeps this operon off. Now finally, in the last condition, when lactose is present, allolactose can be made. And that allolactose binds to the Lac repressor and keeps it off of the DNA. When glucose levels are low, cyclic AMP levels are high. And that cyclic AMP can bind to the cap protein. And now, with cyclic AMP bound, that cap protein can bind to the promoter of the Lac Operon. And in the absence of repression and the presence of activation, here and only here, the Lac Operon will be on, allowing the cell to metabolize the lactose, which is present, and not get distracted with its favorite food source, which is absent. This is how the cell makes a choice, a metabolic choice, a molecular choice, to consume lactose only in the absence of glucose and never at any other time. This choice is wired into the regulation of the Lac Operon. Pretty amazing.